to discouraging the carriage of oxygen in blood, the hemoglobin dissociation curve, oxygen stores in the blood, and their clinical significance and implications. How is oxygen carried in the blood? So going down to Meg.
Two main ways, bound in hemoglobin and dissolved in the plasma.
Fantastic. Actually, you know what, everyone, I'll just get everyone to draw the hemoglobin oxygen dissociation curve just for practice. And then I'll just ask someone to show it up to their camera and we'll kind of go through that. So take a few minutes to do that.
This is the graph that I drew. So this is, does it look backwards to you though?
No, that's great.
Okay. So I drew the oxygen hemoglobin dissociation curve at standard temperatures, so 37 degrees, pH of 7.4 and PCO2 of 40, with a P50 of 26.6, which you can see here. It's a sigmoid shaped curve due to cooperative binding, and it shifts to the right, i.e. there's an increase in P50 with increased hydrogen ion concentration, CO2 concentration, and 2,3 ppg.
Yeah, fantastic. So the first thing, that's a great answer, because you not only, you drew the graph right, I assume you label the axes, I can't remember if I saw that, which I'm sure you did, and then you said the standards. Like, imagine if you're an examiner, what am I going to ask you? Well, what are the standards? What's the P50 and what shifts it to the right? And you kind of given me all of that in, with very little prompting and very quickly. Therefore, I think we move along very quickly as well in the exam. So actually, just tell me, what are the things that shift to the curve to the right again?
Increased hydrogen ion concentration, so decreased pH.
Yeah.
Increased concentration of 2,3 BPG, and increased partial pressure or content or concentration of CO2.
Fantastic. So temperature, did you mention temperature?
I forgot to. No.
That's right. 2,3 dB temperature, CO2 H+, and there's left shift with the opposite, and also carbon monoxide. What is the content? I want everyone to have a go at this, because I feel like this is, again, one of those reasonably tricky things. It can be tricky. So write down how you measure content, or the formula for content, and some numbers in there as well. Take a couple of minutes to do that.
So the oxygen content equation, so it's CaO2, so it's the concentration of hemoglobin. So I've done this for milliliters per litre. So it's the concentration of hemoglobin, which is usually 150 times 1.34, which is the Höfner constant, times the SaO2, so that's all in brackets, close of brackets, plus 0.03 times the PaO2. So for arterial blood, we tend to quote the SaO2 at 97% and the PaO2 of 100, so that gives us 200 mils of oxygen per litre. And then for the venous, we tend to quote the SaO2 at 75 and the PaO2 of 40, which gives us 150 mils of oxygen per litre.
Yeah, fantastic. How much does the dissolved oxygen give you?
Dissolved oxygen?
Yeah.
It's minimal. So oxygen carriage, so it is only 2%, I believe, is dissolved.
What's the formula for how much is dissolved?
Sorry?
What's the formula for how much is dissolved?
I don't recall that one.
No, that's right. And you're right, it's a very extremely small number, and there's so I've got this in mils per deciliter, which just suits what I'm doing here. But so 1.34, the constant, times hemoglobin in grams per liter, times Sats over 100, and your number's right. And then the fact that the dissolved is really such a small amount, imagine a PO2 of 100.
Okay, that's the dissolved portion as the end component of it, okay.
Yes, that's right. I think you actually mentioned that, sorry, didn't you? Because you said 0.3 or 0.03.
Yeah, because I just do it in kiloliters per minute, or you can do that.
Yeah, good. So, great. So you got to the answer. You've mentioned all the terms. You've given suggested numbers as well. So in my example, 1.34 times 15, because it's in grams per deciliter, times the Sats over 100, and then the dissolved portion, 0.003, which is the constant, times PO2, which is 100, which only contributes 0.3 mils. So really, these are small amounts and very academic, unless it's under hyperbaric conditions. What law, Catherine, governs dissolved oxygen content?
So that's the Hoofner's constant. So it dictates that for 1
dissolved.
gram of hemoglobin that's saturated, it can maximally contain 1.34 mils of oxygen.
Yep, so absolutely, that's the hemoglobin, its binding capacity to oxygen, as you mentioned. How about the dissolved part? Which law is that? I keep
Henry's
asking.
law?
Yeah, it is. Yeah, it is Henry's law.
Yeah.
Good. Now, how do you calculate delivery? Next person along. Brian. What is oxygen delivery to you?
Yeah, oxygen delivery, also known as O2 flux, is governed by oxygen content in mils per liter multiplied by your cardiac output.
Okay,
So,
fantastic. So plug in some numbers and get me an amount.
so if we said our normal oxygen content was 200 mils, 100 mils of blood. So that's 2 liters of blood. Sorry, 2 liters of. That doesn't seem right.
That was per liter.
Yeah. Okay. Sorry. 200 mils of O2 per liter of blood multiplied by your cardiac output, which we would say is 5 liters. So it's about 1,000 mils of O2 per minute.
Perfect. And just imagine that. I want you guys to just pick one of these. You're either going to do it per liter or per deciliter, and that way you'll always get this amount of 1,000 mils per oxygen per minute, or as your value. So that was 200 mils per liter times by 5. What happens, Brian, if we gave you your content as 20 mils per deciliter? How do you then get your 5 liters? How do you then get your 1,000 mils of oxygen flux?
Yeah, I'll multiply it by 10. So I generally think of it as per 100 mils, which is why I was quite a bit confused.
No, that's
so
right.
20 mils per 100 mils. So I would just multiply it to get the O2 content to whatever it is per liter, and then multiply by cardiac output, because I think of cardiac output in liters per minute. So.
Yeah, good. Now, so because a lot of times I'm just getting that example that maybe the examiners will give you these numbers with the deciliter amounts. Then you want to multiply cardiac output by 50 deciliters per minute, which is a bit odd, right? 50 deciliters per minute. But really, it's simple maths. If it's 200 times 5, 20 mils per 20 mils per deciliter times 50 also gives you 1,000. So not the trickiest thing, but once you've seen it, it's absolutely fine.
I must admit, even though I agree that's what the textbooks do, I think Brian's way of just multiplying the oxygen content by 10 just definitely makes a lot more sense.
Oh yeah, you're right.
I hate the unit deciliter. Like, I think it's unnecessary.
It's a stupid unit.
Mils or liters, like deciliter is unnecessary.
Yeah. No, I completely agree. That's good. Okay, so this is a good graphical representation, and whenever you do, like what Wes have done here, whenever you actually draw this hemoglobin oxygen dissociation curve, having this dotted line just showing the dissolved portion might be a useful thing as well. But yeah, that's good. Let's keep going. What is the Bohr effect? Next person along. Dan, what do you reckon? Bohr effect.
Sorry, can you hear me now?
Yeah, go for it.
So does the Bohr effect describe the kind of shift in the oxygen hemoglobin dissociation curve with just getting confused between the Bohr effect and the Haldane effect, actually?
Yeah, so one of them refers to oxygen, one of them refers to carbon dioxide carriage. Which one is Bohr?
So Bohr effect refers to oxygen and Haldane refers to carbon dioxide.
Fantastic. And what do you think it's talking about? Is it talking about, yeah, actually, what general kind of thing is it talking about?
So does it refer to kind of what conditions mean that hemoglobin will be more tightly saturated with oxygen?
Yeah, I don't use that term tightly saturated, but I use the fact that you can get more carriage of oxygen. So,
Yeah. So for example, you would get a, like we talked about before, you get a right shift in your curve with things like increasing temperature, increasing hydrogen ion concentration, increasing carbon dioxide, and increasing T3-DPG.
yeah, that's right. And that's the fact that oxygen can be offloaded far more easy in these conditions. So what does that mean? In those conditions, Dan, where is that in the body? Where?
So kind of in the metabolizing tissues.
Yep, that's right. At the tissue level, you get a whole bunch of metabolic bioproducts. You've got high pH, high CO2, and therefore you'll want more offloading. And you can imagine even in exercise, you might have increased muscle temperature. This all facilitates offloading of oxygen, so there's less carriage of oxygen. So that's good. And then the opposite thing will be at the lung level, which makes sense. You just want it to bind more oxygen, so you'll get a relative left shift at the lung level. So what is it? The Bohr effect, and again, a lot of like a lot of these things, whether you're just describing hysteresis or describing perfusion limitation or whatever it is, just get these definitions. This makes the examiner at the very start of your viva, the fact that you can give a sharp definition that's succinct. You know, the first thing you say to them, you want them to have that first impression of you is that, yeah, this person's going to pass the exam. When I'm coaching the second part people, we just talk about getting a really quick summary out there so that it just sounds like they know what they're talking about. Or when they're being asked, how do you assess, how do you assess this patient? You just got to get lots of good facts in quick succession because you want that first impression to be really good. And I've seen this in play. Like I've seen examiners on video asking people who've said really good answers, but at the start they don't say sharp answers. And when they're giving their feedback, they're actually saying that the candidates didn't say stuff, but they had, and it's really quite amazing that this is a thing. And I think it's just natural human nature that first impression, that definition in this exam, that first impression just colors everything that you're going to say afterwards. So hopefully you get your definitions really sharp. The Bohr effect therefore describes alterations in HbO2 affinity from changes in H+ on CO2 concentrations. And like you mentioned, really, it's all about this right and left shift. Why does it happen? I should have asked you that, sorry. So changes in pH affect the numerous electrostatic bonds that maintain the quaternary structure of Hb, and so therefore the stability of the molecule. And this might increase the offloading. Any questions about that? Sorry, before we move on.
Sorry, actually, can I just check? Is it not just the pH, but also the direct effect of carbon dioxide forming Carbamino compounds that alters the hemoglobin's affinity for oxygen?
Now, when they talked about Carbamino compounds, I always thought of that in terms of CO2 carriage, unless I'm missing something.
You're very correct that it is part of the CO2 carriage, but I guess we also say that the hydrogen ions binding to the histidine residues of hemoglobin, that's part of facilitating ongoing carbon dioxide carriage, but it also contributes to the Bohr effect by making a conformational change. I guess just in terms of when we're describing the molecular mechanisms for the Bohr effect, would you say it's just the hydrogen hemoglobin interaction or also the carbon dioxide hemoglobin interaction?
Look, I can't actually remember that, Mason. I'll totally accept that you might be correct there. I assume you got a reference for that, obviously, yeah?
It's probably nuns, I reckon.
Yeah, you could. Hey, if you do find it,
just say that it's the CO2 is affected because obviously that's what's diffusing into the cell and via carbonic anhydrase forming in bicarbonic acid and then dissociating to the hydrogen ions. So if you don't increase the flash pressure of CO2, you won't have the increase in hydrogen ions.
I guess in a sense, Well, I mean, very correct, but I'm also wondering whether the CO2 interacting with hemoglobin directly through forming Carbamino compounds with the terminal amine group, whatever it is, whether that also contributes to a conformational change in the hemoglobin molecule such that it has a decreased affinity for oxygen.
Yeah, if that's a hypothesis there. I don't know the answer to that, but yeah, if you've got that quoted as that also contributes to the Bohr effect, I'll absolutely accept that. I just haven't seen that specific thing written down anyway.
I'll go looking and probably not find it.
Yeah. Hey, and the process of looking and not getting an answer, it's all about getting these connections in your brain. So,
just to quickly jump in, there is a chapter 10, oxygen, in nuns that has a paragraph on this, but I won't read it out because it takes too long.
Oh no, as in if you could take a screen, could you take a photo of that and email it? What's the best way? I'm actually send a email the email that paragraph to me. That'd be great. Oh, here we go. Or we put it in the chat.
I'll try and put it in the chat.
Yeah, try to put it in the chat. Go for it. How long is this paragraph?
It's not too bad actually.
I'm gonna regret this.
Okay. Hopefully that'll work.
Oh yeah, let's download this. Thanks for looking at that. And I might screen share this now, if that works. Everyone can see. The Bohr effect describes the alteration in hemoglobin oxygen affinity that arises from changes in hydrogen ion or carbon dioxide concentrations and is generally considered in terms of its influence upon the dissociation curve, which is exactly what we've said, which is great. Changes in pH affect the numerous electrostatic bonds, which we've said, that maintain the quaternary structure of hemoglobin, which we said, and so stabilize the molecule in the T conformation, reducing its affinity for oxygen. Wouldn't that be just great to knock that one out in the Viva and say words like T conformation? The examiners will be able to remember that. Anyway, similarly, carbon dioxide binds to the N-terminal amino acid residues of the alpha chain to form carbamino hemoglobin, and this small alteration in the function of protein chain stabilizes the T conformation and facilitates release of oxygen from the hemoglobin. So I think that's what you're saying, Mason. So yeah, absolutely right. Awesome. Changing my slides again.
Mark that down as 1 out of 4.
No, that's great.
Yeah,
This is totally a red herring, but isn't the relaxed form with oxygen bound not T for taut?
Yeah, I don't know.
Taut and unbound, yeah.
I don't know that really.
Yeah, that's correct, Rudy. Tense is unbound, so increased CO2 stabilizes it in the unbound form, which you can also think about as kicking oxygen off, which facilitates its delivery to the tissues.
Okay, all right.
Nice one.
And preventing oxygen from jumping on, yeah? So you kind of shift the equilibrium of that reaction. Yeah? No?
So Mason, you guys are all on top of that. Can you answer that?
Yeah, that would be, so I guess I would describe that in the presence of CO2, I would just say it alters the conformation of the hemoglobin molecule such that hemoglobin has a lesser affinity for oxygen and is more likely to release the hemoglobin into the tissues. Sorry, more likely to release the oxygen into the tissues.
It makes sense in my head. Maybe I just won't say it in the exam.
And again, a lot of this, just remember, if you can say this stuff, fantastic. You know, the fact that this electrostatic bond changes due to pH and the quaternary structure, as well as carbamino compounds and the and its effect as well. These all contribute to it. That's fine. Once you're at the point when they're asking you how these things happen and anything beyond that you're doing extremely well. I wouldn't spend a whole bunch of time worrying about that too much, but that's really great. Rudy, can you describe what's going on here? We're talking about CO2 carriage in the blood now.
Sorry, I can't.
Sorry. I'm just trying to make you imagine what I'm putting up here. Great. I think you can see that now.
Yeah, so essentially this is just a representation of the different proportions of which CO2 is carried in the venous versus arterial blood. So in the arterial blood, you have most of the CO2 being carried as bicarbonate, 90%, 5% is Carbamino Compounds, and 5% dissolved. And then in the arterial blood, you have 60% as HCO2, as bicarbonate, and
say that again.
Compounds, which is essentially the Haldane effect, which we were talking about before.
Really, stop you there. Just say that again. So you've just told me what's happening in arterial blood. What's happening in the next bar?
So this is the venous arterial difference. So essentially, this is looking at what happens due to the Haldane effect, or what happens with CO2 offloading the tissues. So an increase in Carbamino compounds, as we were talking about. So I think 70% of increasing Carbamino compounds from CO2 entering the red cell as it diffuses out of the tissues. You have ongoing carriage as bicarbonate, and you have an increased amount dissolved as there's an increased partial pressure of CO2.
Okay, good. Now, the thing that we always get kind of talk about at this point is why on earth are they calling it venous arterial difference with these numbers instead of just talking about venous? Like, you know, on the left bar, we've got arterial, but on the right, we've got venous arterial difference. So, you know, is this, to ask you a direct question, is the amount of bicarb in venous blood 60% of the total CO2 carriage?
Yes.
So yeah, the answer is no, right? Because it's the venous arterial difference that they're trying to highlight here. So you're still going to have a massive amount of total, bicarb carriage. Like this is, your bicarb in the venous blood, it just isn't 60% of this total carriage. That's just not happening. But the difference is accounted for by all of this carbamino compound. So I think I've got a graph here, hopefully. Actually, let's keep going. Next person along.
Sorry, can you just say that again?
Yeah, absolutely.
You're saying that
if I was to ask someone how much percentage of CO2 carriage is bicarbonate in the venous blood? So in the venous blood, how much percentage of CO2 carriage is due to bicarb? The answer is not 60. Because this is talking about the proportions in the venous arterial difference, which is very small. And we're going to hopefully got a graph that shows why that is a problem. Rudy, what is which law governs dissolved CO2?
So it's a gas in a fluid, so it'd be Henry's
law. Do you know the rough amount or the constant?
Yeah, it's much more soluble than oxygen.
Yep.
I think it's 20 times more.
Yep. So give me a number.
Roughly, but I think it's so if Henry's solubility coefficient for oxygen is 0.003, I think it's 0.2.
It's roughly 20. You were right. It's roughly 20 times the 0.003. So, you're not far off if you multiply that out. So Henry's law, 0.060 mils per deciliter per millimeter of mercury. Bicarb is 30%, which is governed by this equation. And the importance of carbonic anhydrase, which we always mention when we talk about this. Liberated H plus binds to reduced HB. And next person along, Luke, tell me about the Haldane effect.
The Haldane effect describes the change in CO2 affinity in blood with changes in blood PO2. It's related to the propensity for CO2 to bind to deoxyhemoglobin, and which has a higher affinity than oxyhemoglobin and permits additional carriage in blood of CO2. And this is applicable in the removal of CO2 from well-oxygenated tissues like muscle, and then helps to offload CO2 at the lungs where the pH is high.
Yeah, fantastic. Talk to me about Carbamino compounds. How does that and the Haldane effect relate?
I must admit, La, this is like, you know, my Achilles heel of respiratory physiology. I just hate this so much. But basically, I understand that Carbamino compounds are structures that exist in blood, mainly inside red cells, that are capable of binding to CO2 and that form through CO2 and hemoglobin formation and increase the carrying capacity of blood for CO2.
Yeah.
So I think that hemoglobin combined CO2, which obviously takes up, allows it to carry it, but then also I think hydrogen ions can bind to Carbamino compounds as well, and that allows the carriage of H+ ions, which can be formed through CO2 dissociation.
Yeah, and look, even though it's your Achilles heel, your definition was sharp and short. You know, deoxygen in blood, increased ability to carry CO2. I mean, that's a fantastic definition. You don't have to go into much detail. What is the cause of the Haldane effect? And you described the relationship of increasing hydrogen ions or CO2, but then the fact that the Haldane effect is contributed to by both that and Carbamino compounds. And then discussing Carbamino compounds, CO2 plus terminal amine groups in the blood proteins due to this equation here, reduce HB, binds more CO2, and the Haldane effect doubles the loading and unloading of CO2 as well. Like again, I feel like I don't really need to understand this at a molecular level to explain it. You know, I just don't think it matters that much. Like there's not going to be a whole viva on this anyway. Yep, so here's the graph, right? So if you think about that, so we saw the left-sided bar graph showing 5%, 90%, 5%, and then we saw the venous arterial difference, which is, you know, which is really just this little portion here. Can everyone see my arrow? Yeah, good. So that expression of the difference was really just what's going on, what's contributing to this difference. Luke, why have they shown it in that fashion? Why show venous arterial difference instead of venous? Just gonna poke your Achilles heel.
Is it just that it's a really small portion of the overall CO2 carriage?
Yeah, and whenever something is small in statistics or in graphs, they magnify it to show what's going on in there. And so why are they doing that?
Well, I guess, you know, in arterial blood, there's about 480 mils of CO2 per litre, and in mixed venous blood, there's about 520. So the difference is only about 40 mils.
Yep, which is not much.
And so if you look at the dissolved fraction here, it's only changing from 5 to 6% on an absolute basis between arterial and venous. But I think the dissolved fraction increases by 10%. The Carbamino compounds, you've seen on this graph, increases from 5% contribution to 9% contribution, but we know that that's, I think, is it 30%?
Yeah, 30%. Exactly, and that's the whole point. It's kind of saying the whole point of, you know, we were talking about respiratory physiology. It's about oxygen delivery and CO2 exchange, respiration and CO2, you know, removal from the body. And then we're saying that, oh, well, we've only got a very tiny amount here that's changed. Like the absolute amount you said was like 40 mils of CO2, but most of that, imagine if we didn't have Carbamino compounds, this would be a more, you know, far less amount. So it's just the body's way of maximizing its ability to get rid of CO2, and a lot of that is due to Carbamino compounds. So that's why they've got it in this weird format. Again, I can imagine someone probing this graph and, you know, probing your knowledge of this graph and asking these kind of questions, because it just seems trivial when I first saw this, but it really just is expressing the importance of Carbamino compounds and the fact that they're CO2 and the terminal amine groups in blood proteins, and then the fact that reduced HB binds more CO2.
Is that the bicarb change, or more importantly, the increased ability of deoxygenated hemoglobin to buffer hydrogen ions contributes to 30% of the Haldane effect, and whereas the Carbamino formation contributes to 70% of the Haldane effect as opposed to the overall arteriovenous difference. I know because I only looked at this last night when doing some other questions.
Thanks, Mason.
But the easier way I remembered was that Carbamino was 30% of the change and 70% of the Haldane, and that added up to 100. And then you could just flip it around and work out the numbers for the other side.
Yeah, nice one. Thanks, Mason.
Is.
It's described really like nebulously in nuns, but I've come to accept it to be the gospel truth.
Okay.
Can I ask a quick question, just if you go back to where you were, just to double check my understanding?
Yeah, go for it.
So if you go back to, that slide. So if you're looking at these two, would you say that the venous arterial difference, you could also call that the additional CO2 in venous blood? So is that why when you go to the next slide, for example, the HCO3, is that the average?
I didn't get the word
by
average.
85%?
The first thing you said.
Well, it wouldn't be because it's closer to 90.
The first thing you said was right. That you could say that this is the extra.
Because I remember being a little bit confused by this when I was learning about it initially.
Yeah.
And I think I saw it written elsewhere as, the additional CO2 in venous blood.
Exactly. Because it literally is amount of venous arterial difference is akin to venous CO2 minus arterial CO2. And that is, arterial plus venous arterial difference equals venous CO2.
Okay.
Which is what you said.
Peter, can I ask you a question on this larger graph that you've now got up now?
Yeah.
The examiners bang on in every one of their exam reports about when doing graphs to indicate the direction of change. So for example, in a graph like this, if you're indicating the Haldane effect is what gets you to move from your arterial point to the mixed venous point. I often find it really difficult because you're often drawing a large graph, but then like an arterial and a venous curve really close together, and then you're trying to put in a tiny little line in between them to indicate difference. Is there anything just from a pure like drawing and exam technique that you use that actually makes it easier to represent direction of change?
Yeah, I never drew this graph because it never needed to come up again. But I don't know what the question will be. I imagine the question will be something like this: draw the CO2 dissociation curve.
And you draw it zoomed in.
Yeah, you could draw that zoomed in. So we might as well go to that now. I think you know the answer to this. It's painful, but let's say for whatever reason you had to draw this whole graph, then you just draw it, and then you know how to quickly draw the CO2 graphs, which is literally just this little segment here, which we're about to draw now. I find this was one of the last things I learned to draw because I just couldn't be bothered, but it's just, just one of those things that you draw it a few times and it's just as easy as the hemoglobin oxygen dissociation curve. So everyone have a go at that, give it a couple of minutes, and I'll ask someone to show me what they've done.
I won't share mine because I want you to tell me if I did it right after just asking you
course.
before. Now, is that backwards or do I need to flip my camera?
No, that's exactly right.
Yeah.
And what are you?
So I've drawn CO2 content on the y-axis and the partial pressure of CO2 on the x-axis. So mils per 100 mils and millimeters of mercury as the units. I've drawn in red the arterial graph, which the key point being with a CO2 partial pressure of 40 and a CO2 content of 48, and then the venous point with a CO2 of 46 millimeters of mercury and a content of 52 mils per 100 mil. The black line in the middle connecting is showing the arterial venous difference. Really, this sort of hollowed point is what you would get if you didn't have the Haldane effect, and so really the Haldane effect shifts you from the bottom curve to the top. And then I've just labelled the oxygen saturation in the two different curves to distinguish them.
Yeah, nice. And so your points are 40 and 46, 48 and 52.
So for 40 and 48 is the arterial, and then 46 millimeters of mercury and 52 for venous.
Yeah, exactly. So just for everyone, when they're drawing this, on the x-axis, this, again, I might be just projecting my own laziness back when I was studying, but this was one of the last graphs I ever drew, and for some reason, it was just painful for me to draw this, but really, I just drew two lines, drew 40, 46, 48, 52, and then just joined some points up, So it was a very easy one to do. So that's good. I'll just reshare my screen. Hopefully that's the one. Yep, so if you were to draw, this is obviously drawing CO2 partial pressure on the x-axis, CO2 concentration on the y-axis, and then just, magnifying that, just as Mason has showed, arterial, mixed venous, 48 content goes to 50, so 48 concentration goes to 52, and the PCO2 goes 40 to 46, and then you've got your PO2 readings as well. And you've done it in saturations, and they've just done it in PO2, which is fine. Oh yeah, so Mason, what is interesting about this process or this graph?
Everything about it, really.
Oh, sorry, it's a beautiful graph.
Do you mind if I clarify the question?
No, you can't. So what's important about it being more of a linear graph than a sigmoidal type graph?
Okay. So, I mean, the relationship is not perfectly linear, but within physiological ranges of CO2, the relationship is effectively linear. I guess, is it related to this significant change in like oxygen uptake doesn't cause significant impairment in CO2 removal? Although it just sounds like I'm making things up now.
Oh, that's right.
Yeah, I'm not sure I can discuss why the linearity or lack of linearity is interesting.
Let's say you had the in the HB oxygen dissociation curve. If you're at the point of PO2 of 200 and you went to 300, how much extra concentration would you have of O2?
Next to nothing, a very tiny amount.
Beautiful. So now I think you know what I'm asking.
Yeah, okay, sure. So as you increase the partial pressure, you have a significantly increased content of CO2 carriage.
Beautiful. It's linear. The lower the HbO2, the more CO2 in this, which you've already expressed, sorry. What the effect of this is a Haldane effect. Why is this H+ in Carbamino, 30% and 70%, which we mentioned before. And I think this is where we're getting to that point. Yep, so great. Now here's the next graph that you hope you don't have to draw, but it's actually not too hard. This lower one here. Mason, describe for me what we're seeing here, this graph.
Sure, so this is a combination graph plotting the partial pressure on the x-axis of either oxygen or carbon dioxide, and on the y-axis the content in mils per 100 mils of, again, oxygen or carbon dioxide. So the lower of the curves is plotting the oxygen curve. Now, obviously it's a compressed scale, but this would realistically be a sigmoid relationship, much like the oxygen hemoglobin dissociation curve.
Yep.
The oxygen content is very closely related to the saturation. The saturated hemoglobin makes up the overwhelming majority of oxygen carriage in blood. The CO2 curve, however, obviously the volume of CO2 carried is much higher, around about 48 mils in arterial blood. And so that's why it's depicted higher up, and it is a more linear relationship within those change. But the important difference between the slope of these two graphs is that for a small change in CO2 partial pressure, there's a large change in the CO2 content, whereas for oxygen, a relatively large change in partial pressure is required to cause, actually, in fact, only a small change in content from about 20 mils per 100 mil to 15 mils per 100 mils.
Yeah, great. And so this is probably a good exercise. Most people are very comfortable drawing the hemoglobin oxygen dissociation curve when it's 0 to 100 of saturations. You could change that 0 to 100, like this region here. You could make that 0 to 20, and that would be the same. And all they're doing is changing saturations on the y-axis to concentration, and that way you can fit the CO2 graph in. So if you're ever asked to overlay, this is your graph. You're really just, like Mason said, you're just shortening your hemoglobin oxygen dissociation curve, and then adding on the CO2 line. And really, here are the points. 48, 52, which equates to 40 and 46 on the x-axis. And what we're saying, it's linear, right? So that's obviously means that the partial pressure going up is in a linear fashion allows for more concentration of CO2 to be held. And the oxygen dissociation curve, after about 40 of partial pressure of oxygen of 40, it just doesn't increase that much, whereas at the lower part of this curve, it does because of its sigmoidal or semi-sigmoidal shape.
Can I ask as well here, this is obviously one of the more difficult graphs they've ever asked us to draw in the past. If you were asked to draw this, would you actually draw two separate curves for CO2 and two separate curves for O2 to indicate the Bohr and Haldane effects?
It just depends what they ask. But I know that, you could just draw
two side by side realistically.
Yeah.
Because when I'd be correct, you can't really plot the arterial and venous points on the same curve.
Exactly. Yeah, you can't, because it's you're assuming different oxygen levels. Again, this is just if the question is asking you to show the difference, I'll draw the previous curve. If it's showing you to plot oxygen and CO2, I draw this curve. It is quite impressive, like what you guys have to go through, or what we all had to go through, which is learning so many different graphs that are slightly related, but for the right circumstance. Anyway, so there is a large increase, 4.7 versus 1.7 mils per 100 mils, CO2 versus oxygen, so that's an interesting point. So a large increase in PO2 is required, versus a large increase in PO2. So just a quick summary, Bohr is all about oxygen, Haldane is all about CO2, and we've gone through that.